Hello everyone. So in this video of mine, I will be discussing the classification of enzymes plus enzymes of clinical importance. So let's start up with, first of all, again a very important topic in enzymes, enzyme classification. Now there were different ways of classification of enzymes, like as per their function, as per the group transferred, as per the moiety transferred. There were so many different ways of classifying. Say for example, the old way of classification was like 2, 1, 1, 1, or you can say 2, or I can say write it 1. So it was a very tedious one. You have to name the enzyme to which class it belongs, subclass, group transferred, subgroup transferred. But as per IUPAC, or you can say International Union of Biochemistry nomenclature, these enzymes were divided into 6 classes. Now one more has been added, so I can say 7 classes of enzymes. And if we talk about the classes, the number 1 class is oxidoreductases, and the mnemonic is OTHLIL. And now since one more class has been introduced, so it will be T. So class 1 is oxidoreductases. Class 2 means T is transferases. H stands for hydrolases, which comes under type 3 or third class, hydrolases. OTHL stands for lyses. I stands for isomerases. Which is type 5, and then comes type 6, which is ligases. And the last, which has been recently introduced, I can say type 7, or it is translocases. So this is how we classify the enzymes into 7 major classes as per International Union of Biochemical Nomenclature. Now this oxidoreductases is considered to be the most elaborative class, as it has 4 subclasses. The mnemonic to remember this is DOOP, D-O-O-P, where D stands for dehydrogenases, O stands for oxygenases, this O stands for oxidases, and this P stands for peroxidases. Or to remember this mnemonic, you can just remember as DOOP. So I can say these are the 4 subclasses of the class oxidoreductases, which is actually first class. Now we are going to discuss each and every class one by one with examples, because again this is very important to understand that enzymes are the backbone of biochemistry. Okay. So first of all, let's take type 1, or simply I would write it as class 1. Okay. Oxidoreductases. Now if we talk about the name, oxidation and reduction are taking place simultaneously, and this is a typical redox reaction. So oxidoreductases is simply catalyzing the redox reactions, and we know that what are redox reactions? The ones in which oxidation and reduction takes place simultaneously. So I can simply define it as oxidation, which is loss of electrons, and reduction, which is gain of electrons. Is taking place simultaneously. So these are typical oxidoreductases class of enzymes, like that. And if we have to, because without examples, again this is not valid. So I can say the example is lactate dehydrogenase. Very important enzyme. Pyruvate dehydrogenase, again a very important enzyme. Commonly we call LDH for lactate dehydrogenase and PDH for pyruvate dehydrogenase. So slowly we will be seeing the reactions also, but since lactic acid is there, lactate and pyruvate dehydrogenase, so I just want to give this reaction. What happens is simple. This is pyruvic acid, CH3COCOH. So what happens is by a reversible reaction, it is converted into lactate, HOH, COOH, and NADH plus H positive will be converted into NAD positive like this. So this is how oxidation will also take place and reduction will also take place. Now just now I told you that oxidoreductases is a very elaborative class and it has 4 subclasses. So the first subclass of oxidoreductases is dehydrogenases. Dehydrogenases means a removal of hydrogen. And if we talk about the examples, we can simply say that the most common example is NAD will be converted into NADH plus H positive. The substrate will come. Along with this, 2 protons and 2 electrons will also come. An example, again, if we talk about NAD, the example is glucose 6-phosphate dehydrogenase, which is a rate limiting enzyme of HMP shunt. This is one example of dehydrogenases. Second example is FAD is converted into FADH2. Again, the substrate is coming and proton, hydrogen ions and electrons. And if we talk about this, you must have noticed this, why I have written NAD is giving rise to, it is converted into NADH plus H positive, whereas FAD is being converted into FADH2. So we never write NADH2, we never write FADH plus H positive. Why? Because this has a low redox potential. And that is why always remember, one molecule of FAD always give one molecule of ATP lesser than what NAD gives. Okay. And if we talk about the example of this, the most important example is succinate dehydrogenase. Right. So this is dehydrogenases class. Then DOOP. So O stands for oxygenases. Now oxygenases, as the word suggests, it is addition of oxygen. So if one oxygen is attached, we call it as monooxygenases. And if 2 oxygen are attached, it becomes dioxygenases. Now again, we have to look for the examples, monooxygenases and dioxygenases. So if we talk about mono, means single oxygen, so example is all the hydroxylation reactions, because it results into hydroxylase. So I can say the examples are tyrosine hydroxylase. Second example is phenylalanine hydroxylase. And third example is tryptophan hydroxylase. And if we talk about dioxygenases, the example of di means 2 oxygen are added. So here the example is homogentisate oxidase, or it is also known as dioxygenase, because sometimes in the exam the question is asked like this also. So always remember homogentisate oxidase, the other name of this is dioxygenase, and the second example is tryptophan pyrrolase. Again, dioxygenase. So these are the examples of monooxygenases and dioxygenases. Now if we talk about the reaction in monooxygenases, then only you will be able to understand the entire reaction. So in monooxygenases, I can simply say that this will be the reaction. Oxygen plus ZH2. So here you can see that only one oxygen is being added up. And if we talk about the product, it is AOH. Hydroxyl group is attached. Plus we can say Z will be given out and water will also be given out. So we can just count the valency also, right? So we can say 2, 1, 3, 2, 1, 3, oxygen is 2, like that. So this is monooxygenases or addition of hydroxyl group here. Then second is dioxygenases. In dioxygenases, we are simply adding one oxygen, so it is a very simple reaction. A plus O2 will give AO. Example I have quoted, tryptophan pyrrolase or homogentisate oxidase, which is also known as homogentisate dioxygenase. Then this is oxygenases. Then second is DOO. So the third class is oxidases. Now there is a difference between oxygenases and oxidases. As you will see, if I write the reaction, then only you will be able to understand this. If I talk about oxidases, I can simply say that it will combine to give oxidized A and it will give water. And if it is in the presence of oxygen, then again A will be oxidized, but along with A it is going to form H2O2, which is ROS. Very, very important. This ROS stands for reactive oxygen species. Reactive oxygen species, which plays a very, very important role in free radicals. Okay. So we can just evaluate. You can see by just seeing the reactions also, this is simple. If we see the product, it is having a hydroxyl group. Whereas oxidation, you can see the oxidized part here. In both the cases, oxidized part is there. This is the basic difference. Or we can say water is also liberated here, right? But the main thing is, this is the oxidized part. And if we talk about again the examples are oxidase, wherever oxidase is written, like cytochrome oxidase, monoamino oxidase, they will come under oxidases class. So it is DOOP. So then the last one is peroxidases. Now, if we talk about peroxidases, it consists of peroxide or catalase, or you can say peroxidases or catalases, right? And we know that they are present in the peroxisomes, peroxide, peroxidases or peroxisomes, so all are mutually interrelated. And if we talk about the reaction of peroxidases and catalases, if we talk about peroxidases only, the example is glutathione peroxidase. Glutathione peroxidase. This is a very, very important enzyme which requires selenium for its activity. Again, this is a very important factor. And what is happening here? we can simply say that H2O2 AH2, it will give twice water plus A will be given out, right? This is an example of peroxidases, and if we talk about catalases, catalases is simply degradation of hydrogen peroxide. So we can simply say that H2O2 will be degraded to form water and oxygen, like this. And always remember one thing, that there is a difference between catalases and reductases or dehydrogenases. Catalases always scavenge the ROS. So catalases always scavenges ROS, which is reactive oxygen species. So this we have to keep in mind. Reactive oxygen species. So this is class 1 of enzymes, oxidoreductases, which is further divided into 4 subclasses: D, dehydrogenases; O, oxygenases; O, oxidases, and the 4th one is peroxidases. Then we come over to class 2, OT. So class 2 is transferases. Now it is clear from the word itself, there is a transfer. Trans. So transfer of anything, be it phosphate, be it amino group, anything which will be transferred, it will come under the class transferases. For example, if I talk about, example can be transaminases, where transfer of amino group is there. Then transketolase, transaldolase, right? And there is one more subclass of this, whenever phosphate group is transferred. So whenever phosphate group is transferred, it is known as kinases. So kinases will also come under this class, and when inorganic phosphate is transferred, it becomes phosphorylase. And transketolase, this is an enzyme again in HMP shunt. HMP shunt kinases is like hexokinase. I'll just try to differentiate between this hexokinase. And if we talk about phosphorylase, the only example is glycogen phosphorylase. So if we have to differentiate between the two, phosphorylase. Okay, so A, B, C, D, and so if we talk about transaminases, simple example which you have to remember is ALT, which is also known as alanine transaminase, or SGPT, which is always bound to rise in liver diseases. AST, the other name is SGOT, which will always rise in any disease related to the heart as well as the liver. So I can say GOT is a cardiac marker and GPT is liver marker. Along with this, other enzymes are also there, but these are the most specific ones. Transketolase, again this requires TPP for its activity, which we will be discussing later. Transaldolase, again it is an HMP shunt enzyme. If we talk about kinases, kinases mein you can say hexokinase is there, where there is transfer of phosphate. And phosphorylase, there is also transfer, but it is of inorganic phosphate. So I just want to differentiate between hexokinase and phosphorylase. So if we talk about hexokinase, we know that here the reaction is simple. Glucose will be converted into glucose 6-phosphate. You can see one phosphate is attached, or the enzyme is hexokinase. ATP to ADP, or magnesium ions are there for this reaction. But if we talk about phosphorylase, phosphorylase has altogether a different function. In phosphorylase, glycogen, which has number of glucose residues, in the presence of this will have glycogen with one residue of glucose lesser, and it is going to release glucose 1-phosphate like this. So you can see that in hexokinase simple attachment of inorganic phosphate is there. Phosphorylase mein transfer of phosphate is there, inorganic phosphate, which comes out like this. That is why glycogen phosphorylase is the rate limiting enzyme of glycogenolysis, and hexokinase is the rate limiting enzyme of this glycolysis, where increased concentration of glucose 6-phosphate starts inhibiting its own production. So this is the basic difference. But whether it is kinase, whether it is phosphorylase, they both are involved in the transfer. Phosphorylase, inorganic phosphate, so I can write it inorganic phosphate also here, just to clarify it. And here you can simply say kinase. So whether it is hexokinase, whether it is glucokinase, galactokinase, fructokinase, they all are different from phosphorylase. There is only one phosphorylase reaction in the body, that is glycogen phosphorylase, right? So this is transferases. And if we talk about SGOT and SGPT, we can simply say that alanine transaminase will simply convert alanine to pyruvate, taking alpha ketoglutarate and glutamate in the presence of enzyme alanine transaminase. And if we talk about AST, simple aspartate will be converted into oxaloacetate, and again alpha ketoglutarate will be converted into glutamate. Same reaction, alpha ketoglutarate to glutamate, but the enzyme would be different, and that is why ALT is known as GPT. In common practice we call them as OTPT, GOT. So OT is AST, PT is ALT. So this is class 2, which is transferases. Then we come over to the class 3 of enzymes. Class 3 is hydrolases. Now it is clear from the word itself, hydrolysis means which catalyzes the hydrolysis, or it breaks the bonds. Whether it is carbon-carbon, whether it is COC, or whether it is CN, whatever bonds are there, it will be broken by hydrolysis. And if we talk about, say, example is, if we hydrolyze the carbohydrates, which bond will be broken? Glycosidic bond will be broken. If we talk about proteins, peptide bond will be broken, which is the basis of all the digestive enzymes, so peptide bonds. If we talk about nucleic acids or nucleotides, beta and glycosidic bonds, beta and glycosidic bond, phosphodiester bonds, there are so many bonds like that. And if we talk about lipids or fats, lipase or an ester bond will be broken. So, and if we talk about the example, all the digestive enzymes will come to the category of hydrolysis. So, example is all the digestive enzymes. Digestive enzymes. Then we come over to the class 4, OTHLIL. So, L stands for lysis here. And if we talk about lysis, lysis is splitting up class. And if we talk about again the examples of lysis, the most common example is aldolase, fumarase, arginosuccinase. You will see everywhere ACE is added. Or we can simply say arginosuccinate lyase, arginosuccinase. Okay, so they will come under lyases class of enzymes. And if we talk about, again we can write decarboxylase also. decarboxylase. So if we talk about aldolase, I'll just show you how it splits up. So if we talk about first reaction, we know that fructose 1,6-bisphosphate in the presence of aldolase, right? It splits up to form 3-phosphoglyceraldehyde and dihydroxyacetone phosphate, which are also linked as isomers with respect to one another. So this is aldolase. Similarly, if we talk about fumarase, fumarase converts fumarate to malate. So this is again splitting up. Then arginosuccinase, this is an enzyme of urea cycle which converts basically, this is again a very important reaction in which this arginosuccinate splits up to form arginine, which further forms urea and giving out fumarate, which is a link to TCA cycle. So here the enzyme is arginosuccinate lyase. So, and decarboxylase, there are so many reactions in which one carbon atom is given out. Whether it is, you can say histidine to histamine, it is decarboxylation. So this is type 4 lyases. Then we come over to type 5 OTHLIL. So fifth class is isomerases. Now isomerases class, we all know isomerism means they are molecular, or you can say they have same molecular formula but different structural formula, right? And isomerases mein there are 3 examples. One is obviously you can say isomerase itself, isomers, or the name of the enzyme is isomerase. Second is racemase, and third is mutase. So these are the 3 examples of isomerases. If we talk about isomerase, I can simply say that example will be glucose 6-phosphate is converted to fructose 6-phosphate. The name of the enzyme is phosphohexoisomerase. Right. If we talk about racemase, racemase means conversion of D-glucose to L-glucose. L-glucose. And if we talk about mutase, mutase mein the most common one is ASE. The enzyme is conversion of glucose 6-phosphate to glucose 1-phosphate, which is a very, very important reaction of in glycogenesis as well as glycogenolysis. And in fact, this is the only enzyme mutase which is common for both glycogenesis as well as glycogenolysis. And the enzyme here is phosphoglucomutase. Similarly, if we have to convert 3-phosphoglyceric acid to 2-phosphoglyceric acid, again here it is phosphoglyceromutase. So this is class 5, which is isomerases. After this, then we come over to class 6, which is ligases. अब ligate करना means to synthesize something, to form something. And here I can say the most appropriate example would be DNA ligase, which we've been studying for the past so many years, which is also known as the glue of DNA. We call DNA ligase as glue of DNA. And another example of this will be carboxylase. Because again we are joining something. So DNA ligase comes under ligases, carboxylase. Again, if we have to write the reactions of carboxylase, I can say that pyruvate will be converted to form oxaloacetic acid. Acetate or oxaloacetic acid, the name of the enzyme here is pyruvate carboxylase. Very important. Similarly, acetyl coenzyme A undergoes carboxylation by the enzyme acetyl coenzyme A carboxylase to form malonyl-CoA. So I'll write acetyl, which is again a rate limiting enzyme also. So these are the two very important reactions of this. Then another important reaction here you can say, another will be which will be an example of this is synthetase. Now always remember one thing, this is just a key point which I am telling you, that wherever we are writing synthetase, that means ATP is involved. They say, for example, glutamine synthetase. Right. What happens in glutamine synthesis? We can simply say that glutamate forms glutamine. ATP is required. And this reaction is strictly irreversible. The name of the enzyme is this. So this is synthetase. Sometimes we write synthase. So the basic difference between synthetase and synthase is that synthetase requires ATP and synthase does not require ATP. Does not require ATP. An example is simple citrate synthase. So many examples are there where no ATP is required and still synthesis goes on. So this is the main difference, and this will come under synthetase, this ligases, synthetase will come under this. Then comes the last class now which has been recently added, which is translocases. Translocases, as it is clear from the word itself, they will help in translocation, movement from one organelle to another. Let's say if we have to write the examples, I can say that potassium pump is there, calcium channel is there, and if we talk about the enzymes, translocases mein, the reaction is endoplasmic translocase. And this is required for the transport, or you can say for the conversion of glucose 6-phosphate to glucose, and the name of the enzyme is glucose 6-phosphatase. So basically what happens is this glucose 6-phosphate, once produced, it is converted into glucose in the presence of enzyme glucose 6-phosphatase. So this is transferred from the cytoplasm to the endoplasmic reticulum. For this it requires this translocase. And again, if this endoplasmic reticulum translocase is absent or deficiency of this leads to glycogen storage disorder, which is type 1b, which actually mimics von Gierke's disease. But here if an infant is born with type 1 endoplasmic reticulum translocase disorder, he is more prone to sepsis. He is more prone to, you can say, infections because of neutropenia. This is the basic difference. Okay. So this is class 7 which has been recently added, translocases. Then another important point after this, here we finish up with the classification of enzymes. But there are two important points which you should know, because most of the students get confused between this. One is kinases, another is phosphatases, and the third one is phosphorylase. there is a difference. Kinases attaches phosphate. Always. Phosphatases removes phosphate. And phosphorylases is phosphorylitic splitting, means the product, your byproduct is with phosphate. Phosphorylitic splitting. And if we talk about the examples, example is hexokinase, glucokinase, fructokinase, galactokinase and so on. And here glucose 6 phosphatase. And here it is glycogen phosphorylase. Right. So these are the three important again terminologies which you should remember. Similarly, you should be able to now differentiate between synthetase and synthase. Synthetase, wherever it is there, synthesis hogi, but with ATP. But when synthase is written, synthesis will be there but without the involvement of ATP, because we are actually focusing on the rate limiting enzymes also here. So here we finish up with the classification of enzymes. Then next, another very important point related to this is what is the difference between rate limiting enzymes and rate committed enzymes, because this is a question which again creates confusion in so many kinds, So what are rate limiting enzymes? Because again, biochemistry without regulation is nothing. Rate limiting enzymes are there and rate committed enzymes are there. Committed enzymes. Now, if we talk about the difference, it is clear from the word itself. Just suppose if you are committed, just suppose if you had taken admission, so you are committed to crack an exam. Okay. Whether it is NEET PG, suppose if you are, planning to become doctors, you have to clear, you are committed to get good marks in NEET UG. Similarly, if you finish your MBBS, then you have to be committed to clear, crack NEET PG exam. So that is a commitment. Okay, rate limiting का मतलब क्या है कि it is limited to certain other things. Once you complete your graduation or once you complete your MBBS, you might opt for some other course. That means you are not only committed to go for NEET PG, you can go for hospital administration and so on. So this is just an example to differentiate between rate limiting and rate committed enzymes. Just say if I talk about rate limiting, okay, example is hexokinase. There are so many examples. I just want to mainly emphasize on hexokinase because we know that hexokinase is catalyzing conversion of glucose to glucose 6-phosphate. Okay. This is the complete reaction. ATP to ADP and magnesium. Now see, this is a rate limiting reaction first of all. Why it is rate limiting? Because glucose, the moment it forms glucose 6-phosphate, it is not limited to glycolysis only. Glucose 6-phosphate has so many other fates, means it can enter glycolysis, it can enter glycogenesis, it can enter HMP shunt, it can enter uronic acid pathway. That means it is not committed to one process. And this is a mandate for every molecule of glucose which is entering inside the cell, it needs to be phosphorylated. That is why it is rate limiting. It is not rate committed. It can undergo glycolysis, it can undergo glycogenesis, HMP shunt or uronic acid pathway. On the contrary, so this is, I can say this is simply rate limiting. And this is an example of feedback inhibition also. Why? Because whenever the concentration of glucose 6-phosphate will increase, it will start inhibiting its own synthesis. But if we talk about rate committed is means it is committed and there is one reaction in glycolysis itself which is an example of rate committed enzyme, and the reaction is fructose 6-phosphate to fructose 1,6-bisphosphate. The enzyme is PFK1, which stands for phosphofructokinase 1. phosphofructokinase 1. And it is actually, a rate limiting step, no doubt, because increased concentration of fructose 1,6-bisphosphate will inhibit PFK. But the most important one is it is a rate committed enzyme. Why? Because fructose 1,6-bisphosphate can only enter glycolysis. Only glycolysis, no other fate is there. Okay, so that means fructose 6-phosphate is committed to undergo glycolysis, whereas glucose 6-phosphate is not committed to undergo glycolysis. It will undergo, but along with glycolysis, it will undergo some other processes also. This is very important, right? So this is another aspect which I wanted to discuss with you people. Now after this, then we come over to another very, very important segment of enzymes, isoenzymes. Because enzymes diagnostically are very, very important again, because they are the ones which are catalyzing all the biochemical reactions. Now coming over to this, what are these isoenzymes? Yeah, how do we define these isoenzymes? Now see, it is clear from the word itself, iso. Iso means similar. So I can say isoenzymes are the forms of the enzyme of the same enzyme catalyzing the same reaction but can be differentiated on the basis of 3 points. One is electrophoretically, immunologically, and structurally differentiated. Means their structure will be different. Obviously, if structure is different, electrophoretic movement will be different, and if electrophoretic movement is different, immunologically they will be different in the body. They will have different type of immune response for different organs. And obviously they will have a different Km, because properties and all, everything will vary. But they are going to catalyze the same reaction, whether they are present in the liver, whether they are present in the brain, kidneys, wherever they are present. So catalyzing the same reaction but differentiated electrophoretically, immunologically and structurally. So examples of isoenzymes are, a very important example is LDH. Now this LDH stands for lactate dehydrogenase. Right. And this is a very important cytosolic marker also, and it catalyzes only one reaction. It converts pyruvic acid to lactic acid. This is pyruvate and this is lactate. Pyruvate, lactate, right? And simply we can say NADH plus H positive or NAD positive. Now the question is, since we are talking about the isoenzymes, this LDH has 5 isoenzymes. Right? And we can simply say that 5 hai, so it is like LDH1, LDH2, LDH3, LDH4 and LDH5. Now the question is, just now I told you they will be differentiated on the basis of 3 parameters. Structurally they are different. Electrophoretically they are different. And obviously, if these two parameters are different, immunologically also they will be different. Now, if we talk about the structure, always remember LDH is tetrameric enzyme, means it has 4 forms. Tetrameric enzyme. So, if we talk about LDH1, this is predominant in the heart, right? If we talk about LDH2, it is predominant in the RBCs. If we talk about LDH3, you can see there is a change in the structure also. It is present in the brain. And if we talk about LDH4 and LDH5, they are present in the liver and the skeletal muscle. Okay. So, if we talk about H4, most important, it is present in the heart. If we talk about H3M, it is present in the RBCs and in WBCs also. H2M2, it is present in the brain. And HM3 and HM4 are present in the liver as well as the skeletal muscle. Up, since their structure is different, that means their movement in the presence of electric current will be different. So, LDH1 is the fastest moving fraction and LDH5 is the slowest moving fraction. The rest are fast, intermediate, medium, like that. But whenever we have to check the serum, whenever we have to check this electrophoretic mobility in the serum, we have to see which one is the fastest moving, which one is the slowest moving. So, if LDH1 migrates faster, that means it is an indication of MI, and if LDH5 is moving faster, it is an indicator of liver diseases. Very, very important. Because LDH is considered to be a marker enzyme of heart also, but nowadays we do not estimate LDH because LDH ke saath the main disadvantage is that if the sample is hemolyzed, because this is a marker enzyme of cytoplasm also, it is going to give wrong results. And this is the only enzyme which takes around 2 to 3 weeks to come back to normal when the person had first MI. So, this is very important. That is why we do not consider it a reliable indicator also, because apart from this, there are other reliable indicators of MI also. So, always remember LDH1 rises in MI, LDH5 rises in liver diseases. Now, here there is one very important thing which is generally asked in the exam. If we talk about a normal individual like we who doesn't have any history of MI and all, always remember LDH2 will be always more than LDH1. But if a person had MI, there will be a change in the ratio, which means LDH1 will increase than LDH2. And that is why this particular ratio in MI is known as a flipping ratio or flipped ratio. Very, very important from examination point of view also. Flipping ratio or flipped ratio. So, in a normal individual, always LDH2 will predominate because LDH2 is present in RBCs, it is present in WBCs in bulk. But whenever a person undergoes myocardial infarction, always remember LDH1 will always show a rise, preferred rise over LDH2. So, this is a very important feature of LDH which has 5 isoenzymes. And another important thing is hemolyzed sample cannot be determined. So, I can just put a note here that hemolyzed sample has to be discarded. Very, very important. To be discarded. Right. So, this is one example of isoenzyme LDH, lactate dehydrogenase, which has 5 isoenzymes. Then second important example of isoenzymes is CPK. The second example is of CPK. Now, what is this CPK? This stands for creatine phosphokinase. Now, we know what is this creatine. We all know that this creatine is an energy reservoir of the muscles. Be it cardiac muscle, be it any muscle, creatine is the one which is a tripeptide actually, which is giving energy to the muscles to contract or relax. So the reaction is basically what happens here. Creatine is converted to creatine phosphate. And the enzyme is CPK here, and obviously ATP will be utilized to form ADP. And one more interesting thing, this creatine is the one which when loses water is converted into creatinine. So most of the students again get confused between creatine and creatinine. There is world of difference. Creatine is the one which is giving energy to the muscles, and creatinine is actually a result. It is an anhydride of creatine which is produced as a result of muscle atrophy. And creatinine clearance test is considered to be the most diagnostic important test for renal function test. It's a component of RFTs basically. So creatinine is an anhydride of creatine which is produced in the body, which is different for males and females. Now coming over to this, since we are dealing with the isoenzymes, so always remember CPK has 3 isoenzymes and it is dimeric enzyme. Di means 2 forms. So it is a dimeric enzyme. And how many forms? 3 isoenzymes. So obviously CPK1, CPK2 and CPK3. And if we have to differentiate between them, again same will be the point. We can say structurally they will be different, electrophoretically they will be different, and immunologically they will be different. Right. So CPK1 is predominant in the brain. CPK2 is predominant in the myocardium, and CPK3 is predominant in the skeletal muscle. CPK1 is the fastest moving fraction and CPK3 is the slowest moving. Now coming over to the immunological importance, CPK2-MB rises in MI, very, very important. And CPK2 is the most sensitive and most reliable indicator in a person suffering from MI. CPK is there, troponin is also there, but CPK is the enzymatic indicator. We all know troponin is a non-enzymatic indicator. So CPK2-MB is the first one to rise in MI. CPK3 is related to skeletal or muscular deformities. And CPK1 rises in anything related to the brain. Ischemia is there, yeah, whatever kind of brain diseases are there, CPK1 will rise. So these are the 2 very, very important examples of isoenzymes, and I will write it here that CPK2-MB is an indicator of, it is a cardiac earliest marker. Very important. So these are the isoenzymes. Then after discussing isoenzymes, then we come over to the last topic in enzymes. What are diagnostically important enzymes? So I'll just give you a list of the enzymes which are actually very, very important. So diagnostically important enzymes. Important enzymes. Now, if we talk about the importance of enzymes, again, let's start up with the liver markers. If we talk about the liver markers, the most important enzyme here is ALT, or which is actually known as SGPT. Very, very important. Okay. So along with this, basically what happens is there are 2 enzymes. One is ALT, other is AST, which is SGOT. So there is a ratio basically AST to ALT ratio, which is also known as AAR. So what happens is, if this ratio is less than 1, that means ALT is more. Okay, so it is an indicator of very important chronic viral hepatitis. Chronic viral hepatitis and non-alcoholic fatty liver diseases. Non-alcoholic fatty liver diseases. And it is always like this in acetaminophen toxicity also. Toxicity. So we can say that whenever there is elevation of ALT, it is an indicator of chronic viral hepatitis. It is an indication in non-alcoholic fatty liver diseases and in acetaminophen toxicity. On the contrary, if we say ratio is more, which means that AST is more. And whenever AST or GOT is more, it is an indication of liver cirrhosis, liver neoplasia, and alcoholic hepatitis. Alcoholic hepatitis. So these are the two very important, and basically these belongs to the class transaminases. So whenever there is any problem with the transaminases, whenever there is any problem in the liver, transaminases enzymes are bound to rise. And these are the two very, very important transaminases enzymes, GOT and GPT. Commonly they are known as aspartate transaminase and alanine transaminase. There are other enzymes also, but this is considered to be the most specific enzymes. Otherwise, we know in liver cirrhosis, in hepatic cirrhosis, there are so many other enzymes which will increase, because liver being the kitchen of the body, it has most of the important enzymes which will rise when any malfunctioning of the liver is there. But we are focusing on the major ones. Then if we talk about, second is if we talk about, say, prostate cancer. In prostate cancer, there are two enzymes which will rise. First of all, you can say the enzyme is acid phosphatase. And the second one is prostate specific antigen, which we all know PSA will always rise. Prostate specific antigen. Third is if we talk about cholestasis. Again, what are the important enzymes which are going to rise? We know that in cholestasis, alkaline phosphatase will rise. But the most specific enzyme which rises in cholestasis is 5' nucleotidase. Most specific. Very important, right? Then fourth one is in acute pancreatitis. In pancreatitis also we always consider two enzymes basically, amylase and lipase. But if we talk about the specificity of this, lipases is considered to be the most specific ones. Up to 5,000 times more than this. Pancreatitis mein. Then next is, this is again very important in alcoholic people or heavy drinkers or chronic alcoholism. In chronic alcoholism or heavy drinkers, the enzyme which shows a rise is GGT. Very important. Gamma glutamyl transpeptidase. Very important. And because it is involved, it is involving the microsomes, that is why. Then if we talk about the enzymes related with bone diseases, the most important enzymes which arises in bone diseases are, the most important one is obviously pre-alkaline phosphatase, or ALP. And I have already mentioned that ALP is a very important enzyme which arises obviously in bone diseases, and this is a marker of obstructive jaundice also. Very important. If a person is suffering from obstructive jaundice, obstruction of the bile duct will be there, and ALP is the one which also rises in liver diseases. Okay, and it works at a very high alkaline pH. It is like 10.9 or it goes up to 11 also. So this is alkaline phosphatase, and we know that bone diseases may there are osteoblasts, there are osteoclasts. So if we talk about bone forming, or we are talking about osteoclasts or osteoblasts, or bone resorption is there. If we talk about this, ALP will increase, and here we can say that in this collagen will also increase, whereas in osteoblasts and osteocalcin will also be there. But in osteoblast there will be N and C telopeptide of type 1 collagen. Type 1 collagen will be there, and similarly C telopeptide of type 1 collagen will rise. So these are the main enzymes which are going to rise in anything related to the bone diseases. And then last but not the least, in acute renal diseases, there are so many enzymes, there are so many factors which arises in renal diseases, but the most common one is osteopontin. So many are there, osteopontin is there, but the most important I am just emphasizing on, and microalbumin. Microalbuminuria, which is the most important and the most sensitive indicator of in diabetes when a person is going in nephropathy. So microalbumin, it is again a very sensitive indicator. Similarly, osteopontin is there, similarly GGT is there, so many are there. So these are the important, diagnostically important enzymes. And then last but not the least, then we come over to the cardiac markers. Now, if we talk about the cardiac markers, there are enzymatic markers, there are non-enzymatic markers also. So the basic difference is enzymatic markers में the most sensitive one is CPK2MB, and non-enzymatic markers में the most sensitive indicator is troponin. So I will just share the chart. So we can just see in the chart, because this is very important from examination point of view. If we talk about CPK2MB, always remember it rises within 4 to 8 hours of MI. It attains a peak value in 24 hours and it comes back to normal in 48 to 72 hours. Like it takes around 2 to 3 days to come back to the baseline. And this is a very important enzymatic marker. And if we talk about the non-enzymatic markers, we can say that it is troponin T, which rises, which is again an earliest indicator, 4 to 6 hours, much before than CPK, attains a peak value same as CPK 24 hours, but it takes around 7 to 10 days to come back to normal. Again, troponin I, 4 to 6 hours, 24 hours, 7 to 10 days. And if we talk about LDH, LDH rises in 24 hours, peak value in 3 to 6 days and comes back to normal in 2 weeks. And then AST, which is none other than GOT, it rises in 12 hours, attains a peak value in 48 hours and it comes back to normal in 4 to 5 days. So I can simply say that the earliest enzymatic indicator is indicator is CPK2MB, very, very important. Non-enzyme indicator is troponin T or troponin I. So these are the cardiac markers. So here we finish up with our topic on enzymes. So in enzymes, what all we have discussed we have discussed the entire enzymes in 3 videos. In the first video, basically I highlighted about the kinetics, the properties of enzymes. In the second video, we discussed about the inhibition of the enzymes, and third में diagnostically important enzymes along with the isoenzymes. Thank you so much.